Well, it's a very great pleasure being here in the home turf of Avram Hershko and Aaron Chickanova. I met them many times, and I'm very stunned by the beauty of the town of Haifa. Reminds me a little bit of California, all of these beautiful flowers, the sea, the climate. So you are all privileged to live in Haifa. I don't feel completely underprivileged to live in New York, but it's certainly very nice to be in Haifa, I must say. I'm going to talk a little bit in the beginning about evolution. Tim Hunt mentioned already that the first cell, I have to correct him a little bit. He said 2 billion years is actually a bit more. 3.5 billion years ago arose. And this cell, or the population of cells that has arisen through a period of tens of millions of years, Earth is about 5 billion years old, has divided ever since, and as Tim pointed out, all present-day cells are derived from this founding population of cells through, if you wish, eternal cell division. So can talk of eternal life, if you wish. Every one of us, from a cellular point of view, is 3.5 billion years old. Now, what you see there is one of the very simple cells first. This one is better. Yeah, this one? Yeah. The technology is still a problem, as I see. Let me take this. This is perfect. Good. Can you put it back? So what you see here on the left is a prokaryotic cell, which is essentially a plasma membrane, and nucleic acids in here, indicated in here. And what happened in evolution, and that is a speculation that goes back to 1980, is that intracellular membranes really evolved from invagination of the plasma membrane, and then of course there is another source of membrane, endosymbiosis of bacteria by other bacteria, and so you get mitochondria and chloroplast, which I don't discuss and which I don't show here. But the important thing is that the invagination of membranes creates things like the nuclear envelope. As you can see here, the DNA is segregated from the rest of the molecules of the cell as DNA becomes larger and bigger in evolution and more prone perhaps to the problems that its location in the cytoplasm would have, exposure to enzymes and so on and so on. It is really segregated in this by this nuclear envelope, and you can see here in modern eukaryotes you form this many intracellular membranes, and the nuclear envelope, which is a double membraned envelope. Now, the big challenge was really to now, once you had segregated the DNA, to get access to the DNA. And so you had to have transport systems that provide you access into the nucleus and out of the nucleus. And this transport organelle is called the nuclear pore complex. Is a very large organelle. It's 25 times the size of the ribosome. The ribosome structure recently having been solved primarily by Ada Jonat here in the neighboring Weizmann Institute. And this structure, the nuclear pore complex, is about 25 times the size. Many of the proteins are insoluble, so they are out of the reach of Kurt Wüthrich. So we have a little bit of time that we can do without being swamped by the functional genomics people. But in any case, the nuclear pore complex evolved from from membrane bending modules. That is, when you invaginate a membrane, you get a very sharp bend in the membrane, and this sharp bend has to be stabilized and is called a membrane coat. And so there may have been a pro-coat in an evolution or pre-coat or whatever you wish to call it, and that then evolved into different coating structures in the cell. As you can see here, the nuclear pore complex is indicated here and then is related to these other coats that you find, coat protein complex 1, 2, and also the clathrin coat that stabilize these bent membranes. Now here you see an electron micrograph of a nucleus, and you see the inner membrane, the outer membrane, and you see here the nuclear pore complex. One of them, here's another one. And you notice that the nuclear pore complex is connected to euchromatin that is transcribed into messenger RNA, and the heterochromatin that is usually not transcribed is in the regions between pore complexes. Now, in yeast, there are about 200 pore complexes, and in our cells, there are about 2,000, and amphibian oocytes have the record. They have 20 million of these pore complexes. And these pore complexes allow bidirectional transport. So transport can go either way, and they are not only transporting proteins and ribonucleoproteins, but also deoxyribonucleoproteins, that is, viruses. And there are about 1,000 transport events per second per nuclear pore complex. So it's a very active organelle. It's very dynamic because it can double in S phase during the small period in the cell cycle, when short period in the cell cycle when DNA is replicated. Also, the nuclear pore complexes are replicated, and one doesn't really know why this is. And then in mitosis, they are disassembled by the kinase cascades that Tim Hunt has described, and then they're reassembled during a telophase. And surprisingly, in spite of the tremendous size of the nuclear pore complex, 100 million Daltons, there are only 30 distinct nucleoporins because there is a great deal of symmetry. Okay. Now, the first biochemical inroad into nucleoporins came when Laura Davis, a graduate student, made monoclonal antibodies against nuclear envelopes and observed this very interesting staining, punctate staining, when she focused on the surface of the nuclear envelope. And when she focused on the equator of the nuclear envelope, she saw this rim staining and also punctate. And that suggested very much that it was an antibody against a nucleoporin. And in fact, we showed this by electron microscopy. And I jumped now a couple of years. Through proteomic approaches, these 30 nucleoporins have been identified, and they are shown to be having a modular structure. And what you have very frequently in yellow here are these arm heat repeats, which are simply antiparallel helices stacked on top of each other to form a superhelix, a solenoid. And then you have many better propellers, and then you have also predicted coil-coil. Also, the structures that we have done isn't a coil-coil, indicating that predictions are not always right. But here is a very important feature of these nucleoporins, and you see them in zigzag indicated here in quite a few nucleoporins that have these so-called phenylalanine glycine repeats. These are about a dozen or so amino acids, and the feature is that they all have phenylalanine, glycine, alanine, and the rest is varied, can be hydrophobic, can be hydrophilic. And these are unstructured fibers that are attached to some proteins that have coil-coil structures or other structures as indicated here. And these FG repeats have been shown to be the docking site of transport factors. So And the family of transport factors that has been discovered has been called karyopherins because they shuttle forth and back between the nucleus. They ferry cargo forth and back between the nucleus and the cytoplasm. And so these karyopherins bind to FG repeats. And there are quite a few FG repeats in the nuclear pore complex emanating, so that you could really look at it as a jellyfish, the tentacles of many jellyfishes, and that sit out there and try to, where the cargo can bind, and then it gets through the central tube of the nuclear pore complex, which is about 40 nanometers in diameter. So very, very big. The ion channel is 100-fold less in diameter. Now here's just a view of the nuclear pore complex. This is a model that Mike Rout made, and you can see the outer membrane here, and here is the channel, the central channel, that is 40 nanometers in diameter, and then this is attached to the membrane via these spokes and rings, and if you rotate this, then you have a bit better idea. You can see the basket here, the nuclear basket in blue on the nuclear side, and then you can see the red fibers here on the cytoplasmic side. These are asymmetric FG repeat structures that are attached to the nuclear pore complex. And you see that as many of the structures are symmetrical, and that you have a two-fold axis of symmetry in the plane of the membrane and an eight-fold axis of symmetry across the nuclear pore complex. And so these fibers, these unstructured fibers, we imagine are moving around, and there is several hypotheses what the consequence of this is. I won't go into various models. We can discuss it subsequently if you're interested. But if you have a protein that doesn't have a nuclear localization signal, it is sort of bounced off here. It can't go through this mesh of tentacles that are moving around. However, if you have a karyopherin, a cap, then the cap can bind to these unstructured filaments and can get easily through the membrane by diffusion, by Brownian motion. And on the other side, there is a nucleotide, there is a GTPase called RAN that dissociates the substrate from the karyopherin. Now here is, can you put a little bit more juice and make it a bit brighter? Because then one cannot, can you make it a bit brighter? Okay, that's good. So here is the structure of one of these karyopherins, which is a solenoid, and it has in one arch, the amino terminal arch, where it can bind RAN GTP, and when the RAN GTP is bound, it puts another loop into the substrate binding pocket so that no substrate can bind. And recently, Yu-Min Chuk, who was a postdoc in my lab, in her own lab, now had a structure where you get the NLS bound, the signal sequence bound, and she has been able to identify certain constant residues and has been able to identify several hundred signal sequences that interact with this particular karyopherin, CAP beta 2. And here you can see a little bit of a rotation. If it does rotate, you can see the RAN, amino terminal RAN binding arch with the RAN GTP in it. You can see this loop that covers the other arch, where the signal sequence is localized. And therefore, when RAN GTP is bound, you cannot bind the signal sequence. And this is a mechanism by which when the cap with its substrate gets into the nucleus and encounters RAN GTP, the substrate is dissociated by the RAN GTP binding. Now, there is another very important structure that has been done by Murray, and what he has shown is the FG repeats indicated here in red dots of these nucleoporins can bind to the concave or convex, sorry, to the convex surface of these arches that I have just shown you, and that 10 of these FG repeats can bind one of the caps, which is indicated here with the substrate. So there is a compaction upon binding of the FG repeats of these fibers to, when it binds to the cap, a very, very large compaction. And this has been experimentally shown recently by Uli Ebi's lab, by Lim Roderick, and they have put these FG repeats on gold chips and did atomic force microscopy and showed when they bind karyopharynx, the caps, that these the size or the length of these fibers changes from 30 nanometers to 10 nanometers. So a very significant reduction. And if you now give RAN GTP to those, the cap comes off and they expand again. So you have a model of reversible compaction and expansion of these fibers, and this is very significant for the transport, to understand transport across the nuclear pore complex. Now, there is, of course, we have to understand much more about the structure of the nuclear pore complex itself. It's a very formidable task. I said 100 million Daltons. And so our idea is to do some X-ray crystallography of individual NUPs or NUP domains or subcomplexes of NUPs. They are very insoluble, so they cannot be done by structural genomics that readily. And so then we hope, and we have experience in that with ribosomes and protein-conducting channels, we try to do a cryo-electron microscopy of the core component of the nuclear pore complex that doesn't have these filaments attached or that have taken away, assemble them from recombinant NUPs, and then project, fit the atomic resolution into the cryostructures. In this way, you can get ultimately you can get an atomic resolution of this very complex organelle. It will probably take us many, many years, but I wanted to give you some progress report of how far we have gone. Initially, we are pretty much alone in this field because the structural biologists said it is utterly boring to do this sort of thing, because what do you find? Better propellers, solenoids, coiled coils, and so they're fit together in some way, but what else is new? I had a pretty tough time to defend myself in front of the Howard Hughes panel, who some of my colleagues said, well, you know, it's sort of pretty boring, but since you have this little halo and since you discovered the nucleopones, go ahead and do it. It probably cannot be done. But I will show you that we have quite some interesting information that we obtained. Now, before I show you the data, I would like to prejudice you a little bit and show you our model, what the symmetric core looks like. We think there are really a series of concentric cylinders. There is the cylinder first of the pore membrane proteins, which we call POMs, that sit only at the sharp bend here, that are integral membrane proteins, only at the sharp bend of between the outer and the inner nuclear envelope membrane. So they're integral membrane proteins. And they interact with the next concentric cylinders that we call the coat NUPs. So these would be the NUPs that coat the sharp membrane bend. And then you have another cylinder that is, we call the adapter NUPs, and finally the channel NUPs, through which the chain, the material goes through. And So if you cut it in a different section through the nuclear pore complex in the plane of the membrane, you can see the four concentric cylinders: the POM cylinder, the coat cylinder, the linker cylinder, and the channel cylinder. And here are the various nucleoporins or POMs that are localized in these four concentric cylinders. And the first one we looked at are the proteins that are in the coat cylinder, here in the blue cylinder, that are the coat proteins, because one of the coat proteins you notice here is Sec13, and Sec13 was discovered as a component of the COP2 complex that is involved in shuttling proteins from the ER to the Golgi complex. And so here you can see the evolutionary relationship that I pointed out in the very beginning, that proteins that are involved in stabilizing sharp membrane bends are actually shared. And interestingly enough, this Sec13 is shared entirely between the nuclear pore complex structure and these COP2 vesicles. They're identical proteins. They don't even bother to alternatively splice. And so here is the haptomeric complex that has first been isolated from recombinant and has been reconstituted from recombinant proteins in Ed Hertz's lab. And you see the various components that are part of this complex. There's a trimer here, a dimer here, and another dimer here, and they form this haptomer. And the protein that we crystallized was this in the middle, the Sec13 NUP145C complex. And this is work of a graduate student, Kuo Zia, Pete Stavropoulos, and Underhölz, a postdoc in the lab. And so this is perhaps too much of a detail, but these are the portions of the proteins that we expressed in E. coli and made a complex out of it and crystallized it, and we got several crystal forms. And in these crystal forms, what we see is a heterooctamer. We see one Sec13 molecule, a propeller, that is invaded. It is a six-bladed propeller that is invaded by the NUP145C, and the NUP145C provides a seventh propeller. This is unique. This does not occur elsewhere, so far as we know, except it was simultaneously found in another protein, in another complex, namely the COP2 complex, where the Sec13 can be invaded by Sec31 in exactly the same way, and the Sec31 furnishes the seventh blade of the propeller. So here you have this just linear drawing of the interacting molecule, Sec13, invaded by this amino terminal of NUP145C, and then it forms a U-shaped structure with in the middle a sharp bend, and then there is an interaction here between these sharp bends, and you have the same structure repeated here once, and then finally you have a dimerization of the two Sec13s here, and so forming you this heterooctamer, which is about almost 300 angstrom in length and is slightly bent, so that we think that this is the entire coat structure that snugly fits onto the coat on the pore membrane domain and coats the pore membrane domain. And the interesting thing is that all the surfaces that are involved have been highly conserved, essentially 100% conserved in evolution. So that these things are not crystal artifacts. Also, we can detect the octamer in solution, and we can also detect it in several crystal forms. So we think it is physiologically significant and represents the real structure. Here's shown again, and you can see the rotation here of this heterooctamer, and you can see at one side when it is rotating that it forms this slightly bent rod, and this bent rod we think is facing the membrane. Now, how do you fit this structure of the haptomer into the ring-shaped nuclear pore complex of the central core? And what we propose is that you have head-to-end association of haptomers, so you have 8 haptomers, and if you do that, you get the right dimensions of the diameter of this outer concentric cylinder, and so this is what we propose, and we therefore propose that you have 4 of such rings in an antiparallel fashion, and here you have our heterooctamer that goes and connects these 4 rings together. So here we found through homology studies that this SEH NUP85 is very homologous, and we think that this forms another column, so that you have altogether 16 columns of similar proteins that form the coat structure. We have to be careful. We don't want to say a solid cylinder. It's more like a fence, which has many holes so that other proteins can go in between and connect the structure to each other. It's a very highly connected structure anyway. And so this is, here's again shown the 2 columns, and here you see the ring that will be formed by these haptomers, one haptomer this way, another haptomer this way, another this way, and so you have 4 rings of 8 haptomers, each forming the coat cylinder. So we got, by viewing the structure of 2 proteins, we got a significant amount of information about the arrangement of these proteins. Of course, I must emphasize that this is, at this point, still a model. There are competing models, but I won't have time to discuss it at this moment. Now, the vesicle, of course, is different than, let's say, the pore. In the pore, you have a concave surface and a convex surface, and in the vesicle, you have only convex surfaces. And of course, it's no wonder then that the SEC13, in both cases, the SEC13 is being used, but the SEC31 that is being used for the vesicle coating is quite different. And again, I won't go into structural things because I'm running out of time. But again, in the SEC13, SEC31, you form an octamer here, and the octamer is forming this lace network of the coated vesicle, where in our case, please don't remember, it's not a solid cylinder. It forms this cylindrical-like structure, not this cage-like structure. Okay, so the next structure that we looked at is structures here of the channel NUPs of the central cylinder, and we looked at the 2 channel NUPs here, NSP1, NUP 49, and NUP 57 in yeast. And mammalian systems, they're called a little differently. This will be slightly confusing. This is work of a postdoc, the 2 postdocs, Ivo Melchak and Andre Hölz, in our lab, and this revealed something very interesting. So again, I must apologize. In the mammalian system, these proteins are called different, NUP 62, 54, 58, and 45. And you don't have to remember all of these, but what is very interesting is that all of these proteins have a central helical region, and they are surrounded by these FG repeat regions. So the central helical regions have been predicted to be coiled, and what we did is these 2 are very similar. As you can see, there's alternative splicing. We made a triple complex, and here is a triple complex of the 3 proteins. But what we noticed when we did sieving, that there is a tremendous, depending on concentration, there's a tremendous variability in the sedimentation, not in the sedimentation, of the sieving of the complex. So it's a very dynamic complex. At higher concentrations, it is in the direction of tetramer and octamer. Now, very disappointingly, when we took the trimer, only the 58 and 45 crystallized, and the other ones it came out of the complex, and only that crystallized, so that we have only the structure for that. And what we found is that it forms a hairpin loop, not the usual predicted coil structure. and with the shorter end and the longer end. And that this loop then, the hairpins aggregate into a dimer, and then this then goes into a tetramer, yeah, let me see, this is a dimer, an octamer, right? And so what we found is that the interface between the two tetramers can slide and can move by 11 angstrom. So that is a new motif, sliding helices. And so we looked a little bit in detail. These sliding helices have a very nice feature. They have very large hydrophilic residues like asparagine, glutamine, glutamic acid, aspartic acid on one side and hydrophobic residues on the other side. And they can slide against each other. And so, we have crystal forms where we have one extreme crystallized and the other extreme crystallized, so that we could reconstruct the sliding that occurs. And so, what we propose here is again showing that these residues that are very large hydrophilic residues are on one side of the helix. And these very large hydrophilic residues can move, and they can therefore sense the space much better, and therefore can slide much better than shorter hydrophilic residues. And so this is a characteristic of these sliding helices. If you want to read more about it, this paper was published in Science last year. And here you see the sliding module of the 58. You can see the helical surfaces here on which you slide. And this, when you arrange these things around the central channel, you can, through this sliding, you can actually you can make the channel bigger or smaller. And this is very important for the function of the central channel, because the central channel has to accommodate very large things like ribosome or viruses. It probably slides all the time, but when a large molecule that goes through, complex goes through the central channel, it can be expanded. And so, so this is a very important new principle in transport that has not been discovered in other transport organelles. You have sliding in channels, but they're maybe 1 angstrom or 1.5 angstrom, but not 11 angstrom for each of these units. So this is a very large sliding. And now there are several of these modules from the cytoplasmic to the nucleoplasmic direction arranged here. And what we think is that you have a large substrate going through. It's like the intestine of a snake. When the snake eats something, the victim slowly moves through the intestine of the snake, and so the very large particle slowly moves through the central, not so slowly, through the central cylinder of the nuclear pore complex. And here is again these other have these units as well, and here you see it a little bit more, so a take-home message for you that the central channel really can expand and can dilate. Now, so where we stand now is that we know something about these NUPs that form the central channel that can expand. And of course, what will be very difficult is to go after the linker NUPs, because they have to absorb this tremendous conformational changes that can occur in the central channel NUPs. And then the coat NUPs are relatively rigid. So we have a very rigid structure, the coat, and then we have linker NUPs which are more flexible, and the most flexible are the innermost channel NUPs. So to come back to my initial slide in evolution, we think that the coat NUPs were the first one to evolve, because the nuclear envelope membrane simply had a very sharp bend, and this was stabilizing. The coat NUPs were stabilizing this bend, and then other proteins were recruited in evolution to form this extremely beautiful and extremely complex and the most complex transport organelle in the cell. which we will hopefully in the next 10 years or so have atomic resolution, at least at the central symmetric core. And that was it. Thank you very much. Oh, I wanted to show you something. Sorry. I wanted to show you. Can you turn it back? I wanted to show you. Can you juice it up a little bit? I wanted to show you some pictures of Dresden. Okay, so this is the skyline of Dresden, of the celebrated skyline of Dresden with the Baroque towers. And on the left side, I cannot see it from here, on the very left side is the cupola of the Frauenkirche. And I'll show you some other pictures. Here is the cupola of the Frauenkirche in its full glory. You can see, can we go back? And this is much better what you do here. Now maybe you can... No, go back. You can see the cupola of the Frauenkirche in this magnificent array of towers of the Baroque time. And now the cupola is shown a little bit in a magnified way. And you can see nuclear pore complexes there, the little windows at the cupola. And you can see how beautiful this whole cupola is and fitting together with the other ensembles there. And particularly beautiful is the lanterna on top of the cupola. And that's what I wanted to show you about Dresden. Thank you very much. The synagogue is a very important modern building. The Jewish community did not want to have the old synagogue back, which was built by Semper, but wanted to have a modern building. And the modern building looks very monumental. It's a cube where the light comes in from the top with tilted walls, and there are gold threads down on these tilted walls, and it is absolutely magnificent. If you go to Dresden, you should see the new synagogue of Dresden. Thank you very much.